Welcome to my channel. This is 9700 May-June 2021 paper 22. The paper which most of Asia did not get right due to the presence of COVID, including myself. Nonetheless, if you are new to this channel, you know what to do. Like, comment, subscribe, and finally, let's watch and learn. Question 1. Part A. Figure 1.1 is a photomicrograph of a region of eukaryotic tissue. Some of the cells are in stages of mitosis. So as you can see over here, the photomicrograph of a eukaryotic tissue is given. And they have said that some of the cells are in the stages of mitosis. What are the stages of mitosis? The stages of mitosis are prophase, metaphase, anaphase, and telophase. Remember the word P-mat. Part 1. Identify which stage of mitosis is shown in cell E and in cell F in figure 1.1. Cell E. It is obviously metaphase because you can see chromosomes are aligned in the middle, somewhat towards the equator of the cell, and that is what occurs in the stage metaphase. Part F, you can see chromatid separation occurring, and that stage is known as anaphase. The chromosomes condense and become visible at prophase. They are aligned in the middle at metaphase. They separate at anaphase. And at telophase, what happens is exactly the opposite of what happens in prophase. The nuclear envelope reforms and the chromosomes become less visible. So, cell E is metaphase, cell F is anaphase, and that will earn you 2 marks. Part 2. Microtubules are present within the cells that are in stages of mitosis, but these are not visible in figure 1.1. State the function of microtubules in mitosis. Microtubules, they form part of the spindle or spindle fibers, as you all know. And what do microtubules that form part of the spindle do? Well, they attach to the centromere. You can also mention that it aids in pulling the chromatids apart. Writing any of these key points will earn you 1 mark. Part 3. State with reason whether figure 1.1 shows a region of animal or plant tissue. Definitely it is a plant tissue. The reason being, cell walls are visible. If you look at this photomicrograph, you can see cell walls. And it has a sort of regular shape, which is not present in animal cells. Animal cells have somewhat of an irregular shape, whereas over here you can see rectangular shaped cells. Therefore, it is definitely a plant tissue. The reasons are cell walls are visible, regular shape of the cells, as well as no pinching in of cytoplasm, which occurs only in animal cells during cytokinesis. So, mentioning it is a plant tissue along with one reason will earn you that 1 mark. If you state it is an animal tissue, it is definitely wrong. Part B. Semi-conservative replication of DNA occurs during interphase before mitosis begins. Write the correct term in the spaces provided to complete each of statements A to D. A. The DNA double helix unwinds and is separated into 2 template strands when hydrogen bonds holding the 2 strands together are broken. Remember, over here they are considering the process known as DNA replication, and initially in a DNA molecule the 2 strands are held together by hydrogen bonds. Therefore, the first answer should be hydrogen bonds, because it is the hydrogen bonds that hold the 2 strands together, which are then broken when it unwinds. Remember that carefully. So what is between those 2 strands? It is hydrogen bonds. Let's look at statement B. One of the template strands of DNA is copied in fragments. The enzyme is required to join the fragments together to form a continuous strand of DNA. Over here, the enzyme which is required to join the fragments together is DNA ligase. If there are fragments and those fragments need to be joined together to form a continuous strand of DNA, the enzyme that is involved in this is DNA ligase. Statement C. Complementary DNA nucleotides are added to the template strands catalyzed by the enzyme blank. So the enzyme which is involved over here is obviously DNA polymerase, because you know that DNA polymerase is an enzyme which is definitely involved in DNA replication. There are 2 types of polymerase, RNA polymerase and DNA polymerase. DNA polymerase is involved in DNA replication. RNA polymerase is involved in protein synthesis. However, since this question is about DNA replication, and it talks about complementary DNA nucleotides are added to the template strands. As you can see over here, I have highlighted the key words which match with the correct answer. So complementary DNA nucleotides being added to the template strands is catalyzed by the enzyme known as DNA polymerase. Statement D. Blank are regions of repeating nucleotide sequences at the ends of chromosomes that allow the continued replication of DNA without the loss of genes. The important keywords over here are ends of chromosomes that allow the continued replication of DNA without loss of genes. And obviously this substance is known as telomeres. Telomeres are definitely at the ends of chromosomes. They are present to allow continuous multiple divisions without any loss of genes. Therefore, the answer over here is telomeres are regions of repeating nucleotide sequences at the ends of chromosomes that allow the continued replication of DNA without the loss of genes. This question is pretty easy and is totally based on the process known as DNA replication. And the enzymes that are definitely involved in DNA replication are DNA ligase as well as DNA polymerase. And the other keywords are pretty much basic. You just have to remember what holds the two strands together. Those two strands are held together by hydrogen bonds, and then you should know what are at the ends of the chromosomes which prevent the loss of genes. Those are known as telomeres. The enzyme that joins the fragments together is DNA ligase. The enzyme that adds complementary DNA nucleotides to the template strand is DNA polymerase. So writing these answers would earn you 4 marks. Question 2. Sugars are transported within phloem sieve tubes from a source such as a mature leaf to a young leaf which acts as a sink. The young leaf also needs water and dissolved mineral ions which arrive at the leaf within xylem vessels. Part A. As the young leaf matures, the quantity of sugar taken up by the leaf decreases to 0, but the need for water increases. Suggest and explain why the quantity of sugar taken up by the developing leaf decreases to 0 over time, but the need for water increases. This question is for 3 marks, and the key parts of this question are: there are 2 types of leaf, a young leaf and a mature leaf. Initially, sugars are transported from the mature leaf to the young leaf. So the mature leaf acts as a source, whereas the young leaf acts as a sink. However, over time, the young leaf will also start to grow. So as it grows, it becomes more and more mature. As it becomes more and more mature, the sugar taken up by the leaf decreases to 0. But the need for water increases, and they're asking for suggestions and possible explanations why this occurs. So the reasons for the decrease in sugar being: the young leaf is changing from being a sink previously to now being a source as the leaf matures. Therefore, the leaf starts to carry out photosynthesis and make its own sugar. As the question itself mentions, the young leaf initially acted as a sink, but now it is acting as a source as the leaf matures. Therefore, what do all sources do? They carry out photosynthesis and produce its own food. In this case, makes its own sugar. Therefore, the reason why the quantity of sugar taken up by the developing leaf decreases to 0 is because of this reason. It changes from being a sink to a source, thus starting to carry out photosynthesis and making its own sugar. Why does the need for water increase? Well, as the young leaf matures or grows, the number of cells increases and there is an increase in size, because obviously as a young leaf grows, there will be an increase in size. An increase in size means there is growth occurring, and growth means the number of cells increases. This therefore means that more cells is equal to more water, as each cell would need water. Increased transpiration occurs, or greater number of stomata present as cell grows in size. You could also mention more cells is equal to more photosynthesis. And what does photosynthesis require? Photosynthesis requires water. So more cells is equal to more photosynthesis, which is equal to more water needed as a reactant. Writing any of these key points would earn you 3 marks. Part B. The figure listed in table 2.1 are present in one or more of the 3 cell types: companion cell, phloem sieve tube element, xylem vessel element. Complete table 2.1 using a tick if the feature is present and a cross if the feature is absent. For simplicity, I shall use present for tick and absent for cross. Let's look at the features. So we have cytoplasm as the first feature. Obviously, cytoplasm is absent in xylem vessel element, whereas it is present in both companion cell and phloem sieve tube element. Therefore, under the column companion cell, you should put a tick, whereas over here I just wrote present. For phloem sieve tube element, it is also a tick, which means cytoplasm is present in phloem sieve tubes. And under xylem vessel element, there should be a cross, which means cytoplasm is absent. Let's look at cell surface membrane. Cell surface membrane is definitely present in companion cell as well as phloem sieve tube element, whereas xylem vessel element doesn't have a cell surface membrane. It has cell walls which are lignified. Therefore, under xylem vessel element there should be a cross, whereas for companion cell and phloem sieve tube element there should be a tick. The third feature, lignified cell wall. Lignified cell wall is only present in xylem vessel element. Therefore, under companion cell and phloem sieve tube element, there should be a cross, and under xylem vessel element, there should be a tick. Nucleus. Nucleus is only present in a companion cell. Both phloem sieve tube element and xylem vessel element, which form part of the vascular bundle, do not have a nucleus. It is only the companion cell which has a nucleus. Therefore, under companion cell, you should put a tick. Under phloem sieve tube element and xylem vessel element, there should be a cross, because the feature is absent. And completing this table as such would earn you 4 marks. Question 3. In mammals, some cell signaling molecules are steroid lipid hormones. These hormones are transported in the bloodstream to reach capillary networks. At a capillary network, hormones pass out of the blood into tissue fluid. Part A. Figure 3.1 is a diagram of a capillary network. First part, describe the differences between the blood arriving at the arterial end of the capillary network and the tissue fluids surrounding the body cells. The question refers to arterial end, and you should know that all arteries carry oxygenated blood except the pulmonary artery. Therefore, the blood coming from the artery would have oxygenated blood. Keep that in mind. So, they're asking us to describe the differences between the blood arriving at the arterial end, which is oxygenated blood, and the tissue fluid surrounding the body cells, which is shown in this diagram. Therefore, the blood arriving at the arterial end would have higher concentration of oxygen than tissue fluid. Blood has red blood cells, whereas tissue fluid does not, as it is too large to pass through the pores of the capillary. Therefore, there is another possible difference. Some other differences are, blood will have lower concentration of carbon dioxide, whereas tissue fluid will have more concentration of carbon dioxide. You could also mention, blood arriving at the arterial end has higher pressure than tissue fluid. Blood will have more proteins, as proteins are too large to pass through the pores of the capillary. And that is also another possible difference, which you could keep in mind. Another key feature is that blood has platelets, whereas tissue fluid has no platelets. Since this question is for 3 marks, just mention 3 differences. What I would choose is ones which are easy to remember, which is regarding the difference in concentration of the gases. So the blood which arrives at the arterial end will have higher concentration of oxygen than tissue fluid. Blood will have lower concentration of carbon dioxide, whereas tissue fluid will have more concentration of carbon dioxide. Writing those 2 points already gives you 2 marks. And for the final point, I would say blood has red blood cells, whereas tissue fluid does not, because red blood cells are too large to pass through the pores of the capillary. Another point that should come into your mind when you think of the pores of the capillary should be blood will have more proteins, as proteins are too large to pass through the pores of the capillary, thus earning you 3 marks. Part 2. Not all the tissue fluid passes back into the blood capillaries to enter the bloodstream. Some of the tissue fluid drains into blind-ended vessels such as vessel X shown in figure 3.1. Name the fluid that is formed in vessel X. As you can see in figure 3.1, vessel X directs the fluid somewhere else. It is not returning to the part shown as blood to vein. And what other structures are present in such areas? Well, the only vessel that should come into your mind is the lymphatic vessel. So the vessels are lymphatic vessels, and hence the fluid in X would be lymph, because the question is asking about the fluid. Name the fluid that is formed in vessel X. Vessel X is a lymphatic vessel. The fluid that is formed in vessel X is lymph. So the answer is lymph. Hormone S is a steroid hormone involved in cell signaling. Figure 3.2 shows a sequence of events that occurs when hormone S enters a target cell. Part B. Explain why hormone S, shown in figure 3.2, does not need to pass through a transport protein to enter the cytoplasm of the target cell. Hormone S can be seen to directly pass through the cell surface membrane. So what does that mean? Well, if it diffuses directly through the membrane of the cell as shown in the figure, then it should satisfy the properties of the molecules that are able to directly diffuse through the membrane. The properties being, it could be a small molecule as well as it could be hydrophobic in nature, because only small hydrophobic molecules can cross the hydrophobic regions of the cell surface membrane. Because if you recall the structure of a cell surface membrane is formed from a phospholipid bilayer, where the hydrophobic fatty acid tails point inwards, that is away from the watery surroundings, whereas the phosphate groups point outwards towards the watery surroundings. Thus, within the cell surface membrane there is a hydrophobic region. Therefore, the properties of the molecules that are able to directly diffuse to the membrane should be that the molecule is a small molecule as well as it is hydrophobic in nature. Since the question asks why does hormone S pass directly through a membrane without the use of a transport protein, you should mention it is a small molecule as well as it is hydrophobic in nature, because only hydrophobic molecules can cross the hydrophobic regions of the cell surface membrane. And hydrophobic means water hating. Remember that carefully. Writing these 2 points will earn you 2 marks. Part C. The target cell can respond to other cell signaling molecules in addition to hormone S. The cell has receptors in the cell surface membrane, in the cytoplasm, and in the nucleus. Explain why hormone S binds only with receptor R in the cytoplasm and not with the other receptors shown in figure 3.2. Each hormone would have a specific receptor that is complementary in shape to the hormone. Remember that carefully. Again, each hormone has a specific receptor, and specificity means that both the hormone and the receptor would be complementary in shape. Therefore, due to the specificity, hormone S can only bind with receptor R and not any other receptor. Since this question is for 1 mark, you just have to mention each hormone would have a specific receptor that is complementary in shape to the hormone. Therefore, hormone S can only bind with receptor R and not any other receptor. The key part of this question being about specificity. Part D. The hormone receptor complex shown in figure 3.2 enters the nucleus and binds to DNA. This switches on a gene coding for a polypeptide that is synthesized in the cytoplasm. Part 1. Name the structure through which the hormone receptor complex enters the nucleus. If you go to this diagram over here, it should pass through the nuclear envelope, and it should pass through the pores of the nuclear envelope. Not just the nuclear envelope, but the pores of the nuclear envelope. Therefore, in order to enter the nucleus, it must pass through the pores of the nuclear envelope. You don't even have to refer to the diagram over here. If you just think about the question itself, the structure through which the hormone receptor complex enters the nucleus. In order to enter a nucleus, it should first pass through the outer surrounding of a nucleus, that is a nuclear envelope. And nuclear envelope has pores in it. Therefore, the keyword over here is pores of the nuclear envelope, earning you that 1 mark. Part 2. Name the processes occurring at B and C. The question clearly mentions that coding for a polypeptide that is synthesized in the cytoplasm. Therefore, this question is about protein synthesis. And if you look at stages B and C in this given diagram over here, B occurs in the nucleus and C occurs in the cytoplasm. And it should definitely be concerned with protein synthesis. Therefore, what are the 2 stages concerned with protein synthesis? The 2 stages we all know are transcription as well as translation. Therefore, B must be transcription because it occurs in the nucleus, and C must be translation because it occurs in the cytoplasm. Again, B, transcription as occurring in the nucleus, C. Translation as it is within the cytoplasm, as well as the question mentioning protein synthesis taking place. Part 3. Name structure G. Structure G in this given diagram over here is concerned with the process of translation. And what could structure G be? Well, the only structure that comes into my mind is the ribosome. Therefore, you can simply say ribosome, or you can even mention ATS ribosome, because the size of ribosomes in an animal cell is ATS. Part E. Cell signaling by hormone S results in the production of a functioning globular protein molecule composed of 3 identical polypeptide chains. After the synthesis of these polypeptides, changes need to occur to form the functioning globular protein molecule. Outline the changes that need to occur to form the functioning globular protein molecule. In order to form a globular protein, it must definitely have a tertiary structure. Therefore, after the 3 identical polypeptides are formed, you should mention about the formation of secondary as well as tertiary structure. Formation of the secondary structure is by folding and coiling to form the alpha helix and the beta pleated sheet. Remember that carefully. When the word secondary structure comes into your mind, you should also think about alpha helix and beta pleated sheet, because they are all interconnected. After this, it then further coils into a 3D manner or a 3-dimensional manner to form the tertiary structure, and that is what the tertiary structure is all about. Folding and coiling in a 3-dimensional manner would result in the tertiary structure, along with presence of bonds holding the tertiary structure together, such as ionic, halogen, hydrophobic interactions, as well as disulfide bridges. Since the question does mention formation of a globular protein, you can also state about the arrangement of the hydrophobic and hydrophilic groups. So the hydrophobic groups would point towards the center. What does hydrophobic mean? Hydrophobic means water hating. Hydrophilic means water loving. So the water hating groups would point towards the center, that is away from the watery surroundings, and the water loving groups would point towards the outside watery surroundings. Therefore, when you think about a globular protein, you should state that the hydrophobic groups point towards the center and the hydrophilic groups lie towards the outside watery surroundings. It is also possible to state other features, such as addition of sugar groups to form a glycoprotein, or removal of the methionine amino acid to form a functional protein. So these are some possible changes that could occur to form the functioning globular protein molecule. Thus, writing 4 of these points would earn you full 4 marks. Question 4. A person who is exposed to tobacco smoke is at greater risk of lung cancer and chronic obstructive pulmonary disease. Many people with chronic obstructive pulmonary disease have both chronic bronchitis and emphysema. These diseases cause changes in the gas exchange system. For example, changes occur in the total lung surface area to volume ratio. Part A. Tar in tobacco smoke has a number of effects on the cells lining the gas exchange system. State the main effects of tar on the cells lining the gas exchange system that are related to lung cancer and to chronic bronchitis. Tar is definitely involved in lung cancer as well as chronic bronchitis. How is it involved in lung cancer? Well, tar is a carcinogenic substance, or a substance which causes cancer. In other words, a carcinogen. This therefore causes mutations, because a carcinogenic substance or a carcinogen would lead to mutations. Mutations occurring would change the sequence of nucleotides in DNA, which could then cause cancer. Therefore, that is how it is related to lung cancer. When you think about the word lung cancer, you should think about mutations. And what leads to mutations? Carcinogens or carcinogenic substance such as tar. So it is all interrelated. And how is chronic bronchitis formed? Tar not only acts as a carcinogen but also paralyzes the activity of cilia on epithelial cells. Cilia therefore does not move to and fro and is unable to carry the mucus up and away from the lungs due to loss of synchronous movement, and hence the mucus accumulates, because function of cilia is to beat to and fro and carry the mucus up and away from the lungs. However, if the cilia is paralyzed, the cilia is not able to move to and fro, and if it doesn't move to and fro, that means the mucus is not carried away from the lungs, and thus it would accumulate. The accumulation of mucus furthermore causes the inflammation of the goblet cells. Remember that key point. Accumulation of mucus leads to inflammation of the goblet cells, which continue to produce more and more mucus, and thus chronic bronchitis occurs. And that is how you explain the effects of tar on the cells lining the gas exchange system. In other words, the epithelial cells. Writing these points would earn you full 3 marks. Part B. A student investigated the effect of surface area to volume ratio on diffusion. Agar was prepared with universal indicator solution and sodium hydroxide solution. The agar was colored blue. 3 cubes A, B and C were cut from a solid block of blue agar. Each cube was a different size. Universal indicator solution changes to a red color in the presence of acid. The student prepared table 4.1 to show the sizes and surface area to volume ratio of each cube. As you can see over here, the table 4.1 is given, the columns being cube, length of each side, total surface area, volume, as well as surface area to volume ratio, which is already calculated over here. Question being, complete table 4.1 by writing the correct units for volume, as well as calculating the total surface area and volume of cube C. What is the unit of volume? Well, how do you find out volume in the first place? Volume is equal to length times breadth times height. So if each of these measurements are in centimeter, centimeter times centimeter times centimeter would lead you with centimeter cube. So the unit for volume is centimeter cube or cm cube. The next part of the question, calculating the total surface area and volume of cube C. In order to find the volume, we already know the formula now. Length times breadth times height. So the length of each side would be the same throughout since it is a cube. So the length and breadth and height is of the same size, that is 3 cm for cube C. So the volume would be 3 times 3 times 3, which is 27 cm cube. How do you find out the total surface area? There's a formula for this. Total surface area is equal to 2 times the whole thing given inside the bracket. Length times breadth plus length times height plus height times breadth. If you look at the formula I have mentioned over here, total surface area is equal to 2 times the whole thing inside the brackets. So length times breadth, that is 3 times 3, plus length times height, that is again 3 times 3, plus height times breadth, which is again 3 times 3. So 3 times 3 is 9. So 9 plus 9 plus 9 would leave you with 27. And 27 times 2 leaves you with 54. Therefore, the total surface area is 54 centimeters square, and thus earning you your 2 marks. Part C. Cubes A, B and C were placed in a small beaker. At time 0 seconds, dilute hydrochloric acid was added to the beaker to cover the cubes. The student timed how long it took for each cube to change color completely. Complete figure 4.1 to show the results that were obtained. Figure 4.1 shows the arrow pointing from shortest time to change color completely to longest time to change color completely. So the order is from shortest to longest time taken. So the shortest time taken would be in the cube which has the highest surface area to volume ratio. As you can see over here, the cube with the highest surface area to volume ratio will take the least time to change color, as diffusion occurs the fastest in that cube. Diffusion is governed by the surface area to volume ratio. Therefore, if there is a higher surface area to volume ratio in a given cube, the faster is the diffusion process. And faster diffusion process means less time taken. So, shortest time to change color completely would be the cube that has the highest surface area to volume ratio. And if you go to this given table over here, the cube that has the highest surface area to volume ratio is cube A. Thus, it should be starting from cube A having the shortest time. Next, it will be followed by cube B, because cube B has the next highest surface area to volume ratio, and lastly cube C, because cube C has the smallest surface area to volume ratio and thus would take the longest time to change color completely. And there you go, your full 1 mark. Part D. Some people with emphysema may be offered lung volume reduction surgery, LVRS, in which diseased lung tissue is surgically removed. One expected outcome of the surgery is an improvement in total lung surface area to volume ratio. suggests why there is an improvement in total lung surface area to volume ratio after the surgery has been carried out. If surgery is not carried out, then the person would continue to suffer from emphysema, which the final result would be pretty disastrous. It leads to alveolar walls collapsing and alveoli bursting, giving lower surface area to volume ratio. If surgery is carried out, however, the removed tissue will contain a higher proportion of diseased alveoli because the surgery itself is about removing part of the lung tissue which is diseased. Thus, if surgery is carried out, the removed tissue will contain a high proportion of diseased alveoli. Therefore, the remaining tissue has healthy and functional alveoli. Hence, an improvement in total lung surface area to volume ratio. It won't be as bad as a person not having surgery. A person not having surgery will lead to the end result being alveolar walls collapsing or breaking down, as well as alveoli bursting. If alveoli bursts, that means the surface area to volume ratio is very low. However, if the surgery is carried out and the diseased alveoli already removed, then there is less chance of alveoli bursting because there will be the remaining tissue which has healthy and functional alveoli. Thus, an improvement in total lung surface area to volume ratio. Part E. In humans, blood that becomes oxygenated in the lungs reaches body tissues without coming into contact with blood that is deoxygenated. Explain how the blood that becomes oxygenated in the lungs is kept separate from the blood that is deoxygenated. In such questions, you should straight away think about the heart. There is a structure in the heart which helps to keep the oxygenated blood separated from the deoxygenated blood, and that structure is the septum. A septum in a heart keeps the oxygenated blood and deoxygenated blood separate. Only oxygenated blood travels to the left side of the heart, whereas deoxygenated blood travels to the right side of the heart. Thus, in order to earn your full 2 marks, you should mention the structure being septum, and you should mention about oxygenated blood traveling to the left side of the heart. Remember that carefully: oxygenated blood, left side; deoxygenated blood, right side of the heart. Thus giving you your full 2 marks. Question 5. Figure 5.1 is a transmission electron micrograph showing parts of 2 plant cells. The function of the middle lamella is cell-to-cell adhesion. The middle lamella is composed of a polysaccharide known as pectin. Pectin interacts with the polysaccharides cellulose and hemicellulose in the cell walls of the plant cells so that the cell walls are held close together as shown in figure 5.1. Part A. Cell structure X in figure 5.1 is a cytoplasmic channel with strands of cytoplasm passing through the cell walls of the 2 cells. Name cell structure X and state one function of this cell structure. As soon as you see the keyword strands of cytoplasm passing through the cell walls, you should definitely get the word plasmodesmata into your head, because plasmodesmata are structures that is formed from strands of cytoplasm that pass through cell walls. Therefore, cell structure X should definitely be plasmodesma because it is singular, whereas plasmodesmata is plural. The question clearly mentions the structure being singular, therefore you have to write plasmodesma. And what is the function of plasmodesma? Their function is to aid in Simplast pathway, which is allowing transport of water between cells. Writing these 2 points, that is a name as well as a function, would earn you your 2 marks. Part B. Researchers have discovered that pectin is synthesized within the Golgi body. Golgi vesicles containing pectin are moved to the cell surface membrane for release. Part 1 suggests why researchers would not have investigated ribosomes as being the possible location for the synthesis of pectin. Pectin is a carbohydrate, it is not a protein, and only proteins are synthesized by ribosomes, whereas pectin, being a carbohydrate, is not synthesized by ribosomes. Thus, the researchers would not have investigated ribosomes being the possible location for the synthesis of pectin, because again, pectin is not a protein. Part 2. Name the mechanism that is used to transport pectin out of the cell. Question mentions Golgi vesicles being involved, and as soon as you see the keyword vesicles, it should either be exocytosis or endocytosis, because vesicles being involved can only mean 2 things, either exocytosis or endocytosis, and the question does mention mechanism used to transport pectin out of the cell, And since out of the cell has been mentioned, it definitely has to be exocytosis, not endocytosis. Endocytosis means taking into the cell. Exocytosis means releasing it from the cell or out of the cell. Juices that are extracted commercially from fruits can be made less cloudy by the breakdown of the cell wall using enzymes cellulase, pectinase, and xylanase. Cellulase hydrolyzes cellulose, pectinase hydrolyzes pectin, xylanase hydrolyzes hemicellulose. Part C. Figure 5.2 is a graph showing the effect of cellulose concentration on the activity of cellulase, which is used in making fruit juice less cloudy. If you go through this graph, as the cellulose concentration increases, the rate of reaction also increases at the start, after which it starts to decline. Nonetheless, there is an overall increase in the rate of reaction. And the question is, describe and explain the curve shown in figure 5.2. Another key thing you should remember is that cellulase acts on cellulose, as mentioned in the question above. So, overall, as seen from the figure, as the cellulose concentration increases, then the rate of reaction also increases. That is the overall trend. For explanation as to why this happens, increase in cellulose concentrations could have led to increase in the rate of collisions between the enzyme and the substrate. In such questions, think about the rate of collisions or about the collision frequency. As there is an increase in cellulose concentrations, it could have led to an increase in the rate of collisions between the enzyme and the substrate, that is, between the cellulase as well as the cellulose. At low cellulose concentrations, it could have been that the substrate was limiting. If you look at this graph, at first, that is towards lower cellulose concentrations, the substrate was limiting and many active sites of enzyme were available, whereas at higher concentrations, most of the active sites are occupied and hence faster reaction. That is a key point which is definitely present in your textbooks. In such graphs where they plot the substrate concentration with the rate of reaction, you should clearly identify what happens at the start and at the end. At the start, it is usually where the substrate is limiting, whereas active sites of enzymes being available, and towards the end, the substrate being available, however active sites being limiting or the active sites being occupied. So as you can see over here, I have mentioned that point. At low cellulose concentrations could have been that the substrate was limiting and many active sites of enzymes were available, whereas at higher concentrations, most of the active sites are occupied and hence faster reaction. You could also mention as to why the curve begins to plateau or become less steep towards the end. The reason being that towards the end, enzyme concentration becomes limiting. In such questions where you have to describe and explain the graph, do mention about the limiting factors at the start and towards the end. Again, how do you describe and explain the graph? The description is basically what the trend is like. And the trend over here is as the cellulose concentration increases, the rate of reaction also increases. For explanation as to why this happens, increase in cellulose concentration leads to an increase in collision rate between the enzyme and the substrate. And you could also describe the graph as such, including limiting factors. Towards the start, there is the substrate being limiting, and towards the end, the enzyme is limiting. And that is why the curve becomes less steep towards the end, thus earning you your full 3 marks. Part D. Ultrasound is one possible method that can be used to destroy microorganisms that contaminate fruit juices. Ultrasound is a term given to sound waves that are out of the range of human hearing. An investigation was carried out into the effect of ultrasound on the activity of cellulase, pectinase, and xylanase used in fruit juice manufacture. For each enzyme, the effect of ultrasound was compared with no ultrasound on the maximum rate of reaction, which is Vmax, Michaelis Menten constant, that is Km, and the catalytic efficiency, that is Vmax upon Km. Table 5.1 summarizes the results. A higher Vmax upon Km indicates a higher catalytic efficiency. The table is given with the enzyme being labeled as cellulase, pectinase, and xylanase. The method is given, comparison of Vmax, comparison of Km, as well as the catalytic efficiency is given. Part 1. In terms of changes in the interaction between enzyme and substrate when ultrasound is used, suggest explanations for the lower Km for pectinase and the higher Vmax for xylanase as shown in table 5.1. As soon as you see the word Km, you should definitely focus on enzyme affinity. Km is linked with enzyme affinity, where lower Km means higher enzyme affinity and higher Km means lower enzyme affinity. There is an opposite correlation between the two. Lower Km means higher affinity, higher Km means lower affinity. So the lower Km for pectinase means that ultrasound has increased the affinity of the enzyme towards the substrate. That is one possible explanation. This could be by making the active site more complementary to the substrate. So after you have identified what has happened to the affinity of the enzyme, whether it has increased or decreased, then you can make out the rest of the answer. If it has increased, that means the active site could have been more complementary in shape to the substrate. There could have been a better fit, a better enzyme substrate complex, whereas higher Vmax for xylanase. Well, if you look at the table over here, higher Vmax for xylanase when ultrasound is used, it could mean that ultrasound increases the collision rate between the enzyme and the substrate. That is a possible explanation. Higher Vmax means faster rate of reaction. As I told you about how Km is related with enzyme affinity, Vmax is related with the rate of reaction. Higher Vmax means faster rate of reaction. Lower Vmax means lower rate of reaction. So higher Vmax for xylanase means that ultrasound increases the collision rate between the enzyme and the substrate. Higher Vmax also means faster rate of reaction, as I mentioned, which could be done by increase in the rate of catalysis after binding. And thus, writing any three of these points would earn you your full 3 marks. However, do make sure you mention a point each about pectinase and xylanase. Part 2. Explain whether the data shown in table 5.1 supports the recommendation that ultrasound can be used in the manufacture of fruit juices. My take over here is that yes, because all enzymes, when ultrasound is used, has higher catalytic efficiency, which does show there is increased productivity when using ultrasound. If you go to the table again, when ultrasound is used, higher catalytic efficiency is shown in all 3 of the enzymes, cellulase, pectinase as well as xylanase. For cellulase, 34 is greater than 29. For pectinase, 945 is definitely higher than 759. For xylanase, again 146 is greater than 125. Thus, higher catalytic efficiency when ultrasound is used. Therefore, yes, it does support the recommendation that it can be used in the manufacture of fruit juices. You could also mention values to highlight this point. Question 6. The diseases myasthenia gravis and HIV/AIDS both involve disorders of the immune system. Part A. The cause of myasthenia gravis involves a response by B lymphocytes. Explain why MG is called a disorder of the immune system. Myasthenia gravis is an autoimmune disease. And what does autoimmune disease mean? It is where the body fails to distinguish between self and non-self antigens, and thus it leads to antibodies being produced which attack the body's own cells, that are self antigens, and that is why it is a disorder of the immune system. Part B. Studies have indicated that T lymphocytes are involved in stimulating the B lymphocyte response that causes myasthenia gravis. Research has been carried out on a vaccine that will provide a person with active immunity against these T lymphocytes and B lymphocytes. Suggest and explain how this vaccine will provide a person with active immunity against the T lymphocytes and B lymphocytes responsible for causing myasthenia gravis. A vaccine provided to the person will contain antigens of the defective T and B lymphocytes, because that is a purpose of vaccine. A vaccine has antigens which are introduced into the body. And what are these antigens? These antigens will be of the defective T and B lymphocytes, because they are aiming to provide a person with active immunity against these T lymphocytes and B lymphocytes that causes myasthenia gravis. So it should definitely be defective in nature. These antigens, when introduced into the body, is recognized as foreign or non-self, thereby stimulating an immune response. In this kind of question, you have to go to it in a flow, starting from what is a vaccine made up of, what happens when these substances are injected into the body, what is the outcome of it. So the person is provided with antigens of defective T and B lymphocytes. This is recognized by the body as foreign or non-self antigens. Thereby it stimulates an immune response, because obviously the body has to defend itself against these foreign substances. Then antigen presentation occurs by the macrophages, which leads to clonal selection and clonal expansion of the lymphocytes. Remember these two keywords, clonal selection and clonal expansion. Clonal expansion is where multiple division takes place by mitosis of the lymphocytes. Lymphocytes are involved with antibody production, and eventually antibodies are produced against these defective antigens. In order to provide a person with active immunity, memory cells should also be formed. So memory cells are also formed in the process. Thus, when the body comes in contact with these defective T and B lymphocytes again, there will be a faster secondary immune response which would occur, and the person is protected from these B and T lymphocytes that can cause myasthenia gravis. Thus, that is how you suggest and explain the process, earning you your full 3 marks. Part C. Many people who are living with HIV develop tuberculosis. If a person does not have any symptoms of TB, one preventive measure is to prescribe antibiotics. This reduces the overall number of cases of TB and deaths from TB. State the disadvantage of prescribing antibiotics as a preventive measure against TB. The main part of this question lies in this last statement which I have highlighted over here. Prescribing antibiotics as a preventive measure against TB. Key thing you must know is that antibiotics should never be used as a preventative medicine but rather as a cure. Else the disadvantage could be that it leads to antibiotic resistance, as the bacterial strain might develop resistance due to mutations occurring within the bacteria, which then makes the antibiotic ineffective. And that is a disadvantage of prescribing antibiotics as a preventative medicine or a preventive measure. It should never be used as such, but it should only be used as a cure. If it is used as a preventive measure, it could lead to mutations. Mutations could lead to antibiotic resistance, and thus the antibiotic could no longer be effective. Part D. Figure 6.1 is a summary of some of the statistics published by Joint United Nations Programme on HIV and AIDS about HIV and HIV/AIDS for the year 2017. The figures shown in figure 6.1 for 2017 are estimated. So as you can see over here, the figure 6.1 shows different kinds of statistics. Globally, 36.9 million people were living with HIV. And the question being, one other statistic published by UNAIDS indicated that in 2017, only 75% of the estimated 36.9 million people living with HIV knew that they had been infected with the virus. With reference to the information in figure 6.1, discuss the importance of this statistic. So only 75% of the whole 36.9 million people knew, which means that around 9.2 million people remaining did not know that they were infected. So those who did not know may not undergo any ART treatment, because out of 36.9 million people living with HIV, 21.7 million people were provided with ART, that is antiretroviral therapy, and after which they do not have detectable levels of the virus in their blood. So those people who do not know may not undergo this treatment, and thus they have a greater risk of dying from HIV/AIDS, as well as a greater risk of transmission because they don't know. So greater risk of dying from HIV/AIDS as well as greater risk of transmission. Since they don't know that they have the disease, they will spread it to others as well. These same people may be sharing needles without knowing and not following the safe measures. Thus, greater risk of developing other opportunistic infections such as tuberculosis. And that is another infectious disease through which thousands of people could die. Therefore, the importance of this statistic is, as mentioned over here, writing any 3 of these points would earn you your full 3 marks. And that is the end of this paper. Hope you found this video useful. If you did, leave a comment down in the comment section below, letting me know your thoughts. Thank you for watching and stay tuned for more.